In the last lecture, we began to discuss the work of Jacques Monod and François Jacob on the expression of lactose metabolizing enzymes in E. coli bacteria and how this work led to the discovery of gene regulatory proteins. I ended that lecture by introducing a hypothesis originally suggested by the physicist Leo Szilard, which is called the negative control hypothesis, to try to describe how this system worked. Let me begin this lecture by reviewing the salient facts and discussing how Szillard's hypothesis appeared to account for them. In normal E. coli, the lactose metabolizing enzymes beta-galactosidase and galactosidase permease, you'll remember, are only produced when lactose is present in the environment. And so we call lactose, in this case, an inducer, inducing the transcription of the genes for these enzymes. Now, Jacob and Monod had described several kinds of mutant phenotypes, two of which clearly represented a loss of function for one or the other of the lactose metabolizing enzymes themselves, fitting our normal view of how mutations affect the expression of gene products. But a third kind of mutation had a very different effect. A mutation in this gene, which they named the LAC-I gene, caused the expression of the two other genes to always be turned on, even if there was no lactose present, suggesting that this gene's protein product must be involved somehow in regulating the expression of the other two genes. Now, Szillard's hypothesis suggested that the LAC-I gene protein acts as a repressor, that's the name he used, on the genes for both beta-galactosidase and galactoside permease, blocking their transcription. In normal cells, Szilard argued, the repressor is always present because the LAC-I gene is always turned on. So beta-galactosidase and galactoside permease are normally not produced because their genes are turned off by the repressor. Lactose, however, Szilard argued, must interact with the repressor in some way, causing the repressor probably to change its shape. Probably, he argued, because lactose would bind to the repressor. And the binding of lactose to the repressor, changing the repressor's shape, would make that repressor no longer able to do its job, which is the way we would imagine proteins can be affected by a shape change. In this sense, then, to go back to the analogy we introduced last time, the repressor protein is the key that must be removed from a lock in order for the enzyme genes to be turned on. What lactose is doing as an inducer is rendering this key itself non-functional, so that the key can no longer interact with the lock. This is a great hypothesis, but how could it be tested? Well, in the late 1950s, an American biochemist named Arthur Pardee came to Paris to work with Monod and Jacob, and it was a collaboration between these three researchers that led to a confirmation of Szillard's hypothesis. The work they did is usually referred to with the somewhat fanciful name of the Pajot-Mo experiment, which is taken from the first two initials of each scientist's last name. Now, the point of their experiment was simply this. They wanted to separate out the effects of the inducer, which is lactose, and the repressor, presumed to be the product of the LAC-I gene, and separately see how they affected the expression of the other two enzyme genes, beta-galactosidase and galactoside permease. Actually, as we'll see, these researchers focused only on looking at the activity of beta-galactosidase because that was an enzyme that they could easily detect using biochemical means, and by looking just at the activity of that one enzyme, they were able to make the general case, they hoped, of how gene regulation was occurring. Now, if they could somehow demonstrate directly that adding the repressor protein to the system turns beta-galactosidase off, then they would have confirmed a basic prediction of Szillard's hypothesis. To do this, Pardee, Jacob, and Monod took advantage of the fact that genes are sometimes transferred naturally from one bacterial cell to another through a process called conjugation. Conjugation is a little bit like sex in bacteria. It's really kind of a bacterial mating process. Now, without going into the details, some bacteria carry an additional piece of DNA separate from the DNA in their chromosome, which we call a plasmid. And they're sometimes capable of transferring that plasmid from one cell to another by one cell attaching a small tube-like connection to another cell. Once that connection is formed between the cells, then the plasmid will travel from what's called the donor cell, the cell that actually reached out with the tube, to the recipient cell. The donor cell is really like the male in this case and is often called the male in bacterial studies, and the recipient cell is like the female. Now, the recipient cell, the cell that receives this plasmid, will essentially have two copies of any genes that happen to be on that plasmid. So this is a way that Pardee, Jacob, and Monod decided to see if they could separately see the effects of the presence or absence of a functional LAC-I gene. Now, they started with mutant bacteria that had both a mutation in the LAC-I gene, that's the gene that makes the presumed repressor protein, and a mutation in the LAC-Z gene. Remember, that's the gene that codes for beta-galactosidase. These mutants they used as recipients in a conjugation manipulation. As donors, they used cells that had normal functional copies of both of these genes, copies that were found on the plasmid. Now, the idea here is that before mating occurs, if you think about those recipient cells, they're unable to actually metabolize lactose because they don't have functional genes for a functional gene for beta-galactosidase. But they also don't have a functional gene for the repressor, so that if they did have a gene for beta-galactosidase, then it would always be expressed because the repressor would be absent. So the recipients are essentially double mutants, lacking a functional LAC-I gene and lacking the gene for beta-galactosidase. Now, the prediction here is a little tricky, and to be honest, Pardee, Jacob, and Monod had to puzzle over their results for some time before they understood them. You would imagine that if conjugation is transferring functional genes for both the LAC-I gene product, that is the repressor, and for the enzyme itself, beta-galactosidase, to the recipient, then when the recipient got those, if that's the conjugation that they did, what you would expect is that the recipient cell, because it now has functional copies of both of those genes, would act like a normal cell. It would be able to express beta-galactosidase, but it would only do so if lactose is present. What they observed was actually something different from this. And actually, in a sense, it was fortuitous that they observed something that wasn't exactly what they expected, because as we'll see, it actually helped them understand the system even better. They found that after conjugation was completed, the recipient cells began to produce beta-galactosidase even in the absence of lactose. Now, they had reason to know that these recipient cells had received both the LAC-I gene, the functional LAC-I gene, and the functional beta-galactosidase gene. So they expected at first, if that was the case, that you would not see beta-galactosidase produced, but it was produced without lactose. But here's the key. It was only produced for a couple of hours at most. If you measured the amount of beta-galactosidase being produced by those recipient cells after conjugation, production would occur maybe a few minutes after conjugation began, as expected if there were no repressor. And production of beta-galactosidase would continue for maybe 60 minutes, 90 minutes, or 2 hours. But then the production of beta-galactosidase would begin to tail off and eventually go to zero. And after a couple of hours, these recipient cells now behaved like normal cells, because beta-galactosidase would be normally turned off, but if you added lactose to the system now, it would turn beta-galactosidase on. This is puzzling, and if it puzzles you, it puzzled them, and it puzzled me for a little while when I first heard it. But here's the key to understanding this, something that they figured out using a different piece of information. And this is the information they found. The transcription of these two genes occurs at different rates. In other words, for reasons we can't go into at the moment, one of these genes is transcribed quite quickly, whereas the other gene is transcribed relatively inefficiently. Which gene was which? The gene that was transcribed very quickly was the gene for beta-galactosidase. So when conjugation occurred and the recipient cell had that functional beta-galactosidase gene, it began to make copies of the enzyme very quickly. And you would see that in the production of the enzyme. But the LAC-I gene was transcribed less efficiently and more slowly. And so at first, the LAC-I protein, the repressor, wasn't present in the cell in any quantity. But over time, 90 minutes or a couple of hours, the LAC-I protein product began to build up. And as the LAC-I protein product built up, the production of beta-galactosidase was turned off, exactly as you would predict if that protein product of LAC-I was a repressor, acting as a regulatory protein that was turning off the production of beta-galactosidase. Well, in 1961, Jacob and Monod published a summary model of how they thought gene expression is controlled in this system based on the Pajot-Mo experiment and a number of follow-up experiments they did after that. Let me describe this model to you because it really provides the first and in some ways the simplest model for how one protein could act to regulate gene expression of another protein. Now, genetic mapping studies had shown that the functional genes involved in lactose metabolism, that is the gene for beta-galactosidase, galactoside permease, and a couple of other genes involved, actually all occur in one long sequence immediately adjacent to each other on the molecule of DNA. What they proposed, as we know is the case now, is that immediately preceding this sequence of the coding regions of those genes was a single promoter, a promoter where the RNA polymerase would bind. But in between the promoter and the coding regions for these genes, they proposed that there must be a regulatory sequence of DNA, which they called the operator. The operator was where the repressor protein produced by the LAC-I gene would bind to the DNA. Because the operator was physically located between the promoter and the coding regions of these genes, once RNA bound to the promoter, it simply couldn't move on down the line and transcribe the RNA products of the DNA of the coding region. Now, how does lactose fit into this model? Well, as was originally suggested by Szilard, lactose, we know, does bind directly to the repressor protein, and in so doing causes a conformational change, a shape change, which makes that repressor protein unable to bind to the operator. So here's how the system is thought to work. Normally, the repressor is always present in the cell. The LAC-I gene, by the way, occurs at some other region of the genome, not too far away, but not as part of this group of the promoter, operator, and the functional genes themselves. So LAC-I is normally producing the repressor protein, which is normally sitting on the operator. And although RNA polymerase is always trying to sit on the promoter, it simply can't move down the line. Well, when lactose gets into the system, it binds with that repressor protein, and the repressor protein is taken off the DNA, and RNA polymerase can move on. So as we suggested, the repressor is a lock that has to be removed from the key, is a key that has to be removed from the lock, and the way this happens is by lactose making that key non-functional. Now, we know that there are many other operon I should say, I'm sorry, I got ahead of myself there. Jacob and Monod referred to this whole system together as an operon, a unit centered around the operator region, which was the region that controlled activity of the gene. And they called this lactose metabolizing system specifically the LAC operon. Now, we know now that there are many other operons in bacterial genomes, each responsible for regulating the coding of functional groups of genes involved in many other metabolic processes or cell functions that the cell has to carry out. And that in general, it's the binding of these regulatory proteins to the operator regions of these operons that controls gene expression through negative control. As we'll see in a moment, however, the way gene expression works in eukaryotes is different from this. And there are a couple of differences I want to point out right away. First of all, in eukaryotic cells, Genes in general all have their own individual promoters and other regulatory regions. That is, each individual gene is controlled by itself. Genes are not controlled in a group. Second, the specific molecular mechanisms responsible for how expression is turned off and on in operons, that is, the binding of a protein to an operator that blocks the movement of RNA polymerase, is actually unique to prokaryotic cells. That doesn't occur in eukaryotic cells. As we'll see, the way regulatory proteins affect the activity of RNA polymerase in eukaryotes is usually more subtle and more complex than this, involving helping that RNA polymerase to find and bind to the promoter in the first place. Nonetheless, the work of Jacob Monod and their colleagues was an important breakthrough because it demonstrated a mechanism whereby one protein could turn on and off the expression of other proteins by controlling transcription. Now, there's one more point I need to make about how prokaryotic operons work that I haven't mentioned yet, but it's a point that makes a transition to our next topic, which is to take a quick look at how gene transcription is controlled in eukaryotic cells and how that differs from prokaryotes. The interactions between the repressor protein and the operator, and between the inducer, lactose in the case of the lac operon, and the repressor, are not the only factors that determine how gene expression occurs in operons. In addition, there's another regulatory region that is also adjacent to the promoter, to which another regulatory protein binds that actually helps the RNA polymerase find the promoter in the first place, to bind to the promoter in the first place. We didn't mention this because I wanted to simplify the system to make the general point, but as it turns out that this way of regulating gene function, by having proteins bind to regulatory regions that affect whether or not RNA polymerase binds or not in the first place, is the major way that eukaryotic cells turn genes on and off. Well, there are a number of reasons why gene expression should be different in eukaryotic cells and prokaryotic cells, mostly all having to do with the fact that the genomes of eukaryotes are larger and more complex than the genomes of prokaryotes. Remember, prokaryotes have a single circular chromosome. It's not very big. Eukaryotic genomes may be orders of magnitude larger and are broken up into many different, sometimes many different linear chromosomes. Eukaryotic genomes are also different because they have large stretches of DNA that don't directly code for proteins. These are stretches such as the introns we saw that have to be removed from coding regions of genes. There are also large stretches of repetitive DNA that appears to have no obvious function, that just occurs in the genome. Finally, another major difference between the way that eukaryotic and prokaryotic genomes work is that eukaryotic DNA, you'll recall, is packaged up quite tightly in a bunch of protein, forming a structure we called chromatin. In other words, bacterial DNA is naked. Eukaryotic DNA is enveloped in a protein structure. It's this fact that eukaryotic DNA is enveloped in the protein structure that brings us to the first major difference we have to consider in how gene expression works in eukaryotes. But we can only consider this briefly now because we really don't understand much about how this aspect of the system works. If the DNA of eukaryotic cells is normally enveloped in proteins, how can RNA polymerase get to work on it in the first place? Somehow, the DNA in a eukaryotic genome must become unpackaged from the chromatin in order to be exposed so that RNA polymerase can do its work. How that happens is the subject of very active research. We know that it does happen by a variety of means. We know that the regions of DNA that are being transcribed in eukaryotic genomes do become unpacked from chromatin. We don't know exactly how that works, and more importantly, we don't know how it's regulated. It used to be that we thought that the function of chromatin was largely just structural support, a good way to package and protect the DNA, all of the DNA that has to be crammed in a eukaryotic cell. Now we're beginning to believe that the chromatin actually plays its own regulatory function, that the process by which pieces of DNA are unpackaged from its enveloping proteins are a way that gene expression is turned on and off. Stay tuned for how that works. However that occurs, once the DNA is exposed, then we have the same problem we had with prokaryotes, and that is, how do we control whether or not a particular piece of DNA is transcribed by RNA polymerase or not? We know that not all exposed pieces of DNA will be transcribed. There are additional levels of control, and that's where we want to go to next, to review the basic mechanisms that eukaryotic cells use briefly. We're not going to have time to talk in detail about these mechanisms or to give you the experimental background that led us to this understanding, but I want to give you a flavor for how gene regulation occurs in eukaryotes. I mean, we're eukaryotes, many important organisms are eukaryotes, and more importantly, when we begin to look at the process of development in a few lectures, these mechanisms of gene expression will become very important to us as we understand how gene expression varies as an organism develops. Well, let's very briefly go back to our analogy of locks and keys to get a bearing on where we are and where we want to go. Each gene in a eukaryotic genome, in the broadest sense, is associated, as we've said, with three kinds of regions of DNA. Remember, there's a coding region, and in the case of eukaryotic cells, that coding region can be quite complex because it can have, as part of it, a number of introns that have to be removed. This is the part of the gene that will actually contain information, ultimately, about the structure of proteins. Each gene, as we would now expect, also has a promoter associated with it. And this is a sequence of DNA where RNA polymerase will come down and bind and eventually use as a landing pad to start its work in transcription. Finally, each gene, as we would expect now, has associated with it one or maybe several regulatory regions. And as we've seen before, these regulatory regions are like locks, and there are regulatory proteins that bind to these regions, just like keys. Now, an important thing to keep in mind is that the binding of these proteins, regulatory proteins, to the DNA, to the regulatory regions, is usually highly selective. In other words, there are particular sequences of bases on the DNA that will interact only with the structure of particular kinds of proteins. And just like the key you use in the car, which in this case has a particular series of teeth that will influence the position of a particular series of tumblers in the lock, there's only one form of the protein in general that will interact with a particular region of DNA. Now, in eukaryotes, as I've said, it's the efficiency with which RNA polymerase binds to the promoter of a particular gene that will ultimately determine whether that gene is transcribed or not. And here's the key point, it's the binding of regulatory proteins to regions around that promoter that will determine how well the RNA polymerase will be able to bind. Now, as you can imagine, the system gets pretty complicated because there are dozens of proteins that can be involved in what you might think could be a relatively simple process. As we'll see later on, the fact that there are many proteins involved in the process actually gives us a higher level of control, kind of a hierarchical level of control, which can be very useful for the way that we would want to see gene regulation occurring. Another difference between gene regulation in eukaryotes and gene regulation in prokaryotes is that there are actually a number of proteins involved in the process, proteins necessary for RNA polymerase to get started, that are really kind of generic proteins that would be used to turn on any gene. We call these generic proteins basal transcription factors. These proteins must bind with the promoter and with RNA polymerase, and in fact they build up to produce something that we call a transcription initiation complex. In other words, a number of these generic proteins need to conspire or collaborate with RNA polymerase just for RNA polymerase to get its job done. Now, in prokaryotic cells, RNA polymerase is pretty self-sufficient. Once it gets rolling, it more or less can move down the line and transcribe on its own. But in eukaryotic cells, RNA polymerase is essentially moving along with an entourage of proteins, all of which are assisting in its work. But these proteins, these basal transcription factors, as I said, are generic. They're not what's turning on and off the transcription of a particular gene. Each gene will also have associated with it then what we would call the regulatory transcription factors. These are the specific proteins. These specific proteins, as we would guess from what we've seen so far, will bind with particular regions of the DNA, regulatory regions. They might also bind with some of those basal transcription factors and collaborate with the whole transcription complex as it moves along. So we have not only protein-DNA interactions and the specificity therein that helps regulate genes in eukaryotes, but also, in some cases, protein-protein interactions. It's all about locks and keys. We just have a lot more keys, and now we have some different kinds of locks. As we saw for prokaryotes, regulatory transcription factors bind to regulatory regions of the DNA that are adjacent to the promoter. That is, you can find a promoter region and just maybe as part of it or right next to it on the gene, you'll find a region which is the region where a regulatory transcription factor might bind. But here we see another interesting difference as compared to prokaryotes, because there are also regulatory regions in eukaryotic genomes associated with a particular gene that might be occurred at some distance away from that gene on the DNA molecule. In some cases, up to 20,000 base pairs away. These more distant or distal regulatory regions, as they're called, were first discovered only about 20 years ago. And they were called enhancers at the time. They were called enhancers because somehow a particular regulatory protein, when thrown in the mix, would enhance transcription of a gene. But it wasn't clear how it could do that because it was known that the region of DNA with which that protein interacted was nowhere near the gene that was being transcribed. It's easy to imagine in general terms, given what we know about protein structure and function, how a transcription factor that is binding right near a promoter, right near where it could interact with RNA polymerase or the other basal transcription factors in the transcription initiation complex. It's easy to imagine how that could affect the function of the whole system. Because the proteins are right next to each other, they can interact with each other and in so doing change each other's shapes. But how could an enhancer located 20,000 base pairs away from the DNA have an effect? Well, the answer seems to be that the DNA actually bends around back on itself, bringing the enhancers, which are interacting with their regulatory regions, in direct physical proximity to the promoter of the gene that's being regulated. In other words, the DNA is twisting over backwards, bringing different regulatory regions from quite different areas all into this one little place. It does get complicated. And if you study the molecular biology of gene regulation, there are dozens and dozens of details, different proteins that you have to keep track of to understand specifically how the system works. But again, it's all about locks and keys. Regions of the DNA where RNA polymerase has to bind, where regulatory proteins have to bind, and the presence or absence of those regulatory proteins that ultimately will turn on and off whether a gene is going to be expressed or not. In some cases, those regulatory proteins will have a positive effect by binding to the DNA or to the transcription complex. They make it more likely for the gene to be transcribed. In other cases, they have a negative effect. By binding, they make it less likely that RNA polymerase is going to be able to bind or the transcription complex will be able to do its job. But whether or not proteins are present, regulatory proteins, is what determines whether other proteins are going to be expressed. Now, in the next lecture, we're going to look at another aspect of how cells get particular proteins that they need to do the job. Not whether a protein is present or absent in the cell, but whether the protein is actually located in the right spot in the cell. which is another problem that we have to solve.